nanometer-sized liposome in 3 different ways. First is we try to figure out the structure and integrity of nanometer-sized liposome during air-jet nebulization. Second is we are looking onto the size. As I told, it's an important parameter to consider for aerodynamic properties of this particle. And third is what are these aerodynamic properties of liposome once they are airborne. So I will start one after the other. The first one is structure and integrity of nanometer-sized liposome during atomization. So here is our hypothesis. We have this liposome particle, and this small triangle, red triangle, represents the drug molecules. And when this drug molecules is being atomized through an atomizer, which I will illustrate a little slides to follow, it can lead to leakage of this drug and change in the morphology. So the question we are asking ourselves is what is the mechanism of leakage? And once we know the mechanism of leakage, we can employ this to minimize the drug leakage in this kind of characterization. So we chose four different lipid composition to compose the liposome, and these lipid composition had been taken from our understanding of the molecules which are available in human lung. So it eradicates the bioavailability and biodegradability issue. And once we take this 4 composition, we employ in a systematic way. There is no systematic study known to us which has reported that the composition effect of different components can affect the leakage of this liposome. So what we do is we have taken 2 lipid ingredients which are available in the human lung, and we have added some additives like cholesterol and PEG, which are known to influence the membrane integrity, but a systematic study was not available to our understanding. And these are some TM images which shows that lipids of low charge, these 2, the blue represents the neutral charge lipid. If there are neutral charge lipid, there are multi-layer like structure which are visible by HRTM images, whereas in case of the PEG, the fibrous bristles can be visible in a dry HRTM mode. So we studied the leakage in following. So this is an atomization. I will just briefly state that atomization includes the reservoir where the liposome suspension is being taken, and then there is a liquid feed tube from where the liquid is being sucked up, and it is being contacted with a high pressure airstream. The droplets which are generated at this particular point is a micrometer size droplet, which is called as primary droplets. Roughly the scale, if you put some numbers, the scale is few hundreds of micrometer, and they're smashed on this baffle, resulting in generation of secondary droplets which are called as respirable droplets. Typically size range is 1 to 10 micrometers. So there where we are putting the particles, we are just feeding those droplets with liposomal particle, and we are collecting by a conventional aerosol collection device called as impinger. Here there is a high velocity airstream, and at this point, because of the high velocity, the particles are being stripped off from the air line, and we characterize the leakage. Now this is a graph which shows the percentage leakage on the y-axis. On the x-axis there are different nebulization parameter. We have taken this small r, PR represents the pressure and T represents time. Now pressure, if we increase the upstream pressure, obviously the impaction or the shear experienced by this liposome at this point is much higher. And if we increase the time, there is this possibility that the liposome can get recycled within the nebulizer. So it experiences the shear force again and again, leading to more disruptive effect. We have put our experimental data in an experimental matrix design, and the typical value of pressure is taken, low pressure is taken as 10 psi, high is 30 psi, time is first 10 minutes, and then time is 30 minutes of nebulization, and these figures are being taken what is being used in clinical delivery of drugs. So we observe that there is, we have taken this for one concentration. If we increase the liposome number concentration, the leakage pattern essentially remains the same, which gives us a partial conclusion that vesicle rupture and not fusion could be a potential mechanism of leakage. And you can also observe when we add cholesterol, the leakage is contained within 12%, even at high conditions. So it gives us a conclusion, which is of course known in other literature for micrometer size vesicle, that cholesterol addition can minimize the leakage. So coming to the second point, which is the size distribution and morphology of the liposome generated by 2 different methodologies. So we know that up to now that air jet nebulization have shear effect. So can we employ some other novel generation technology which are known for aerosolizing bacteria with low shear? So here comes electrohydrodynamic atomization, another aerosol generation system which is not known for liposome up to now. So what we do is this electrohydrodynamic atomization is basically we have a capillary where the liposome suspension is being fed, and at the tip of the capillary, due to surface tension and electrostatic force, it forms a cone. At the tip of this cone, a pinch of droplet is being taken and entrained is using airflow stream. Now this is another aerosol generation technique, so we compare this with a conventional conventional air-jet atomization or nebulization technique. So electrohydrodynamic atomization gives us a possibility of generate liposome of generate droplets of 130 to 200 nanometer size, which is comparable to liposome. So we hope we can eradicate any aggregation of the liposome in a droplet and have a fine control. The results are following. We observe over here that when we have liposomes, it's essentially prepared in a saline buffer. So even we don't have any liposome, the dry droplet is going to end up in a residual particle. So when we have the zero lipid mass concentration, which corresponds to zero liposome number concentration, there is a polydispersed droplet size which is generated, which is represented over here as dry particle size. Whereas as far as the electrohydrodynamic atomization, the distribution of the droplet as well as the size of the droplets are much less. So as we go on successively increasing the liposome number concentration, we see one peak comes one peak starts showing up in the particle size distribution, whereas in electrohydrodynamic atomization there are two peaks. What we did is we pulled out the particles at these peaks and tried to investigate the morphology to get a better understanding why there is a single peak for air-jet upon aggregation. We concluded the observation using the TEM images as inclusion of multiple particles in a droplet would lead to an aggregated pattern of liposome. Whereas in case of electrohydrodynamic atomization, though they are generating nanometer sized droplet, but the two different pattern of aggregation, one is head to tail. The tail of one liposome get aggregate to the head of the other liposome, as well as side by side aggregation. So there are actually two different agglomeration pathways which result in two different size distribution.